Good afternoon, class. So this is going to be chapter 2, part 2 online lecture. So we're going to pick up where we left off. So we were talking about the periodic table, and we were talking about chemical bond formation. So remember, I stated last time that the whole purpose for chemical reactions is so that atoms, valence electrons can become in their most stable form, okay? So when atoms have incomplete valence electrons, and remember the valence electrons are the outer shell electrons, okay? So when they have an incomplete outer shell, incomplete valence electrons, okay, that's what drives chemical reactions, because they're trying to, the electrons are trying to reach the most stable form, okay? And a lot of times that's going to be by transferring electrons or sharing electrons. They can reach the most stable form by doing one of those two things. All right, so there are two types of bonds that electrons will create if they share their valence electrons, okay? When you share electrons between two atoms, that is called covalent bonds, okay? And covalent bonds is a good way. We're going to talk about electron negativity in a few slides, okay? But essentially, when atoms have about the same electron negativity, meaning, look at it this way: if you have 7 valence electrons, and remember 8 is the magic number, atoms want to have 8 is a complete set of valence electrons. So if you only have 7, for instance, valence electrons, okay, versus an atom that has 1 valence electron, that atom with 7 valence electrons is going to be very electron negative, meaning it's going to be able to pull that 1 electron from that atom that only has 1 valence electron, and that's going to give that atom that had 7, now it will have 8, okay, and it's going to be negatively charged. And then the atom that gave away its 1 electron is going to be positively charged, and it's not going to have any electrons. And that's essentially, those are ionic bonds, okay? Those are ionic bonds. Things like sodium, chlorine form ionic bonds, okay? So those are transferring electrons, and that's because one, in that case chlorine, if you're doing sodium chlorine, one atom is very highly electron negative, meaning it's able to pull that electron from the other atom easily, okay? But when you have atoms that are both about the same strength in electron negativity, okay, then one atom isn't strong enough to pull the electrons from the other. So when it's not strong enough to pull the electrons from the other, you form covalent bonds. They end up sharing their electrons, okay? That's why they form covalent bonds, because they're not strong enough to actually pull the electrons from each other, so they end up sharing them. All right, so for instance, they're showing you on this slide, okay, you have 2 atoms of hydrogen. Remember, hydrogen has 1 electron, 1 valence electron, okay? So remember, I talked about electron negativity. These hydrogens are going to be about the same electron negativity, so they don't have, one or the other hydrogen isn't strong enough to pull the electron from the other atom, okay? So what they're going to do is they're going to share. So if you look right here, H2 right here, and you count how many valence electron, that's 1, 2, and you count 2 for both, okay? So even though it's in the middle, you count it as 2 for each hydrogen. So each hydrogen now has 2 electrons, and now remember, for hydrogen's orbital, that is a full set, and so hydrogen is happy like that. Now let's look at oxygen. You got 1, 2, 3, 4, 5, 6 you have six. Well, oxygen, you have 6 valence electrons, okay? And then you have another oxygen atom with 6 valence electrons. So remember, 8 is a magic number. 8. So oxygen wants to have 8 valence. It wants to have a complete set of valence electron, which is 8. Okay. So in order to do that, you're going to form this double bond. So now you count the electrons that are being shared. So the electrons in the middle, okay, both of them can count those. Okay, the ones in the middle, both of them can count those. So that's 1, 2, 3, 4, 5, 6, 7, 8. That's 8 on the for the oxygen on the right, and then you go to the oxygen on the left. You count the ones in the middle, can be counted for both. 1, 2, 3, 4, 5, 6, 7, and that's 8. So now both oxygens have 8 valence electrons. It's happy. It has a full set. So remember with this double bond, so this is called a double bond, the electrons in the middle are the ones that are being shared. You can count those electrons for both oxygen, okay? And then the ones that are surrounding the oxygen by itself, you have to count that for that individual oxygen. So that's how that works, okay? All right, so this is just showing what we got through showing. So remember, you have 2 nitrogen atoms in this case, all right? So remember, obviously, these are 2 of the same atom. So the electron negativity for both atoms is going to be the same. So one atom isn't going to be strong enough to pull the electron from the other. All right, but look at the valence electron. Remember, the valence electron are the electrons in the outer shell. So how many valence electrons does nitrogen have? That's 1, 2, 3, 4. That's 5. So this nitrogen has 5 valence electrons. And this nitrogen has 5 valence electrons. So it needs 3 more. It needs 3 more in order for it to have 8. Okay. So what's going to happen when nitrogen chemically react with each other is going to form N2. Now let's count the valence electrons. And remember, we count the ones in the middle can be counted for both nitrogen. So we got 1, 2, 3, 4, 5, 6, 7, 8. That nitrogen on the left has 8, a full set. We have 1, 2, 3, 4, 5, 6, 7, 8 on the right as a full set. And this is called a triple bond, okay? So this nitrogen right here has a triple bond, all right? But you see how you count the covalent, you see how you count the electrons that are being shared in the middle, okay? All right, so you have molecular formula. You have structural formula, which is a notation used to represent atoms and bonding. Okay, so that's just showing you that this is a single bond. Okay, if you had 2 lines, that's a double bond. If you have 3 lines, that's a triple bond. Okay, but then you also what they usually write them as, you see molecular formula. Okay, where they just have H2. So they're not actually showing you the bonding. They're just showing you how many hydrogen atoms there are. Okay, so again, covalent bonds can form between different atoms or between same atoms. But remember what I said before, okay? Covalent bonds generally form between atoms that have very close electron negativity, meaning neither atom is strong enough to pull the electrons from the other. So if they can't pull the electrons from the other, the next best thing is to share. Okay, so they share because they're not strong enough to pull them from the other atom. Okay, all right, so this is electron negativity. It's what I already talked about. So electron negativity is an atom's attraction for electrons in a covalent bond. All right, so let me also say this, okay, so you don't get confused. So even in covalent bonds, okay, so I talked about electron negativity in reference to an atom pulling electrons from another, and that is true. but you can also have in a covalent bond, you can also have what's called polar covalent bonds, meaning that yes, it's a covalent bond, they are sharing electrons, but even in that covalent bond, one atom is more electron negative than the other atom. So it's pulling, even though they're still technically sharing electrons, but one atom is pulling the electrons closer to itself. It's more electron negative than the other atom. Therefore, those are called polar covalent bonds. And one of the most common polar covalent bonds that we talk about is water. Okay, so remember, electron negativity is an atom's attraction for the electrons in the covalent bond. Okay, so it could be in a covalent bond, and it could be just, you can use the term electron negativity in general. Okay, essentially, an atom that attracts electrons stronger than another atom is more electron negative than the other atom. Okay, so even when you're sharing electrons in a covalent bond, and water is a perfect example, it's a very polar molecule, because even though, see, we don't have an image of it. Yeah, here we go. Even though you have H2O, so you got two hydrogen and an oxygen, okay, this is a covalent bond. However, oxygen is way more electron negative than the hydrogen. So oxygen is way more electron negative than hydrogen. So even though they're sharing the electron, oxygen is actually pulling the electrons closer to itself. So the oxygen is actually pulling those electrons closer to itself. Therefore, within this polar covalent bond, oxygen is slightly negative and the hydrogens are slightly positive. And I can't stress this enough. This nature of this polar nature of water is what drives a lot of the characteristics of water. For instance, normally a molecule this small, when it evaporates, remember water evaporates at 100 degrees Celsius, okay? Normally a molecule this small, okay, if it was non-polar, it would evaporate at way lower temperatures, okay, because it's a very small molecule, okay? So it would evaporate at way lower temperatures than 100 degrees Celsius, but because water is polar, okay, and essentially what happens is this hydrogen is positive, the oxygen is negative, another water molecule is the oxygen is going to be attracted to that hydrogen, and then another water molecule, the hydrogen is going to be attracted to the oxygen, and another water molecule is going to, the oxygen is going to be attracted to the hydrogen, and so forth and so forth. So when you add up all those, they call them hydrogen bonds. When you add up all those hydrogen bonds, okay, they're weak, but when you have millions and millions of them, it actually becomes a relatively strong force, okay, even though it's a weak force. But when you add up all those polar hydrogen bonds, what happens is water now evaporates at 100 degrees Celsius as opposed to a very lower temperature for a molecule of that size, okay? And that's very important because water being able to absorb, so water is also an energy sink, okay? The sun rays can strike and warm up the water, and water can actually absorb a lot of the sun's energy without boiling, essentially, okay? And then once it gets hot enough, once it gets 100 degrees Celsius, of course, it's going to boil and it's going to evaporate, okay? But it's because of the polar nature of water that water boils at 100 degrees Celsius, essentially, okay? For a molecule that size, it would normally boil or evaporate way lower temperature than that, okay? So that's a very important nature for water, okay? So yes, water is a covalent bond, but it's a polar covalent bond, okay? Oxygen is pulling the electrons closer to itself than the hydrogen, and the oxygen is slightly negative and the hydrogens are slightly positive, okay? And that allows water to form hydrogen bonds between each other. All right, so then you're also going to have what's called non-polar covalent bonds, okay? In this case, in a non-polar covalent bond, the atoms share the electrons equally. Okay, so carbon dioxide is an example of a non-polar covalent bond, and you see carbon dioxide, the electrons are shared evenly. That means carbon isn't pulling the electrons closer to itself than, and oxygen isn't pulling the electrons closer to itself. So in a non-polar covalent bond, they are sharing the electrons evenly between each other. All right. All right. So covalent bonds are actually considered a strong chemical bond. Okay, but there are weaker chemical bonds, such as ionic bonds and hydrogen bonds. Okay, I just talked about hydrogen bonding between polar water molecules. Okay, so I already talked about hydrogen bonding between polar water molecules. And again, remember I said the oxygen is slightly negative, hydrogens are slightly positive. Remember, opposites attract. So one oxygen in a water molecule is going to be attracted to a hydrogen and another water molecule, okay, and that's going to keep going on and on and on and on, and then you add up all those hydrogen bonds. That makes water have its characteristics of why it boils at 100 degrees Celsius as opposed to boiling at way lower temperature, because water is a small molecule. Usually small molecules like that will boil at a way lower temperature, but it's because of the hydrogen bonding between water, all right, that it boils at 100 degrees Celsius. All right, very important. And then ionic bonds is when you have like sodium and chlorine. All right, so remember chlorine has 7 valence electrons, sodium has 1. Okay, so remember the chlorine is going to take the 1 electron from sodium. Okay, so that chlorine now has 8. And it's going to have a negative charge because it gained an electron. And then the sodium is going to have a positive charge because it no longer has an electron. When you get rid of or you don't have an electron, you become positive. Okay. So that positive and negative between the sodium and chlorine is what an ionic bond is. Okay. So that's considered also a weak chemical bond. All right, so this slide is talking about hydrogen bonding, and I've already talked about it. Okay, so I'm going to move on from this slide because I feel like I've already talked about hydrogen bonding enough. But again, between things like covalent bonds that are polar covalent bonds, okay, you have hydrogen bonding between a positive atom and a negative atom, okay? So I would advise you watch the animation on hydrogen bonding in this YouTube video, okay? A lot of times seeing an animation, seeing a picture helps put everything together. Also, so we already talked about the fact that hydrogen bonding is one of the reasons why water boils at 100 degrees Celsius, okay? But another instance that you can see how water sticks to each other, okay, due to the hydrogen bonding strength, is this water strider that's walking in a pond, okay, because of the high surface tension of the water due to its hydrogen bonding, okay? So that's, again, another example of properties of water due to hydrogen bonding. So here's this summarizes ionic bond, complete transfer electrons, sodium, chlorine. The positive and the negative charge is what forms the ionic bond. It's the opposites attract between sodium and chlorine. Okay, a polar covalent bond. Remember, one atom is more electron negative than the other, so it's pulling the electrons closer to each other. The one with the electrons pull closer to itself, it's going to be negative, partly negative, slightly negative, and the hydrogen is going to be slightly positive. Okay, and then you can form hydrogen bonds between another water molecule that has a hydrogen slightly positive and this slightly negative oxygen. And then you have non-polar covalent bonds. The electrons are shared equally. All right, so again, we already talked about ionic bonds, okay? And that's when you have an atom that is way more electron negative than the other atom. And in that case, it's able to pull, essentially strip the electron from the other atom because it's way more electron negative than the other atom, okay? And the one that receives the electron becomes negatively charged, and the one that gave away its electron becomes positively charged. All right, so here's sodium, and remember we're looking at the valence electron, the outer shell electron, and sodium only has one electron in its outer shell, and chlorine has 1, 2, 3, 4, 5, 6, 7. Okay, 7. So chlorine is way more electron negative, meaning that it's way stronger. Its electron attraction is way stronger than sodium. So chlorine is going to be able to strip sodium of that one electron and become negatively charged. And then the chlorine become negatively charged, and the sodium will become positively charged. So this is just showing you that the chlorine is stripping sodium of its one valence electron, okay, because it's way more electron negative than the sodium. And then sodium now, since it gave its electron away, now has a positively charged, and the atom that received the electron is going to have a negatively charged. Okay, all right, so just nomenclature. So in nomenclature, the atom that is positively charged is going to be called cation. All right, cation. And think of the T for positively charged. The T is crossed. It kind of looks like a plus sign. Okay, kind of looks like a plus sign. So think of the cation for positively charged, and then the negatively charged ion is called an anion. Okay, an anion. And just in general, okay, just know that, and this goes even when you're dealing with your battery in your car, red is traditionally represented for positive. Okay, all right, so red is traditionally represented for positive. Also remember that. All right, so chemical reactions are the making and breaking of chemical bonds. So just understand that, again, this isn't a chemistry class, but you remember the law of conservation of mass. Matter cannot be created or destroyed. So what's really happening during chemical reactions, you aren't creating or destroying matter. All you're really doing is rearranging atoms, okay? So during the chemical reaction, all you're really doing is rearranging. You're breaking bonds and forming new bonds. Okay, so in this case, you have two molecules of hydrogen gas plus one molecule of oxygen, and you form two water molecules. Okay, so what happened during this chemical reaction is you broke bonds and formed new bonds. Okay, but you still have the same number of atoms. You still have the same number of hydrogen atom, which is 4. 1, 2, 3, 4, and the same number of oxygen atom, which is 2, 1, 2, but they're in different chemical bonds. Okay, so remember, you're not, with chemical reactions, you aren't creating or destroying matter. All you're doing is rearranging atoms, essentially. And remember anabolic steroids. That's build stuff up. Catabolic, break things down, and the sum of your anabolic and catabolic reactions is metabolism, okay? All right, so again, we talked about the way that chemists write compounds is by molecular formulas, okay? It's just a shorthand, kind of short notation. You know, in a water molecule, H2O, there are two hydrogen atoms and one oxygen atom. And this is sugar. This is glucose, C6H12O6. You have 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. Okay. All right, so then you have different types of chemical reactions. Synthesis is going to involve taking, it could be A plus B plus C, but in this case, we just have A plus B, keep it simple. Taking two individual atoms and then creating one, okay? So this is synthesis. You're combining atoms and molecules to form a larger, more complex molecule, okay? And again, anabolic, this is during the anabolic process. You're gonna do synthesis reactions. All right, so this is showing you have individual amino acids, and then you are going to bind the individual amino acids together to form a protein, okay? That's a synthesis reaction. All right, so decomposition reaction is the opposite, catabolic. In this case, you're breaking things down. So you're gonna take a more complex molecule and break it down into smaller molecules. So AB broken down to A plus B, okay? So this is glycogen. This is one of the storage forms of pretty much glucose is what the glycogen is, storage form of glucose. And in this case, you're gonna break down glycogen when your body needs glucose. It's low on, your cells need sugar. It's gonna break down glycogen, a storage form of glucose, into individual glucose molecules, okay? All right, and this is catabolic. All right, so then you also have exchange reactions, or also called displacement reactions. So in this case, it can involve both synthesis and decomposition. But essentially what happens is you're kind of switching partners in this case in exchange reactions. So you have AB plus C, now you have AC plus B. All right, you're kind of switching partners. All right, or you already got something like this. AB plus CD gives you AD plus CB. Okay, those are all displacement or exchange reactions. All right, example of exchange reactions are ATP transfer its terminal phosphate group to glucose to form glucose phosphate, okay? So ATP gave one of its phosphates to glucose to form glucose phosphate, okay? Exchange reaction. All right, so again, one common, which I'm sure you've seen this chemical reaction before in all of your previous biology classes since middle school, actually probably since elementary, but photosynthesis, okay. Remember, plants can make their own food, but photosynthesis is a very, very important chemical reaction because remember, all the energy on the earth, its primary source is from the sun, okay? But we have to harness that energy, okay? So plants are considered primary producers, all right? They're able to harness the energy from the sun and turn it into a chemical energy form, which is sugar, okay? So they take the energy from the sun, using photosynthesis, and they use carbon dioxide plus water plus the sun energy to create sugar, and it gives off oxygen as kind of a waste byproduct. Okay, but essentially, the energy from the sun has been transformed into a chemical energy form in the form of sugar that your cells can now use that sugar to create ATP. Okay, and ATP is, think of it as the energy currency of the cell. Whenever the cell needs energy, it's going to use ATP for that process. Okay, so photosynthesis is very, very important because without producers harnessing the energy from the sun, and then other animals eat the plants, and then other animals eat the other animals, and so forth and so forth, and you create your food pyramid. You wouldn't be able to sustain life. Well, there wouldn't be any higher order organisms because higher order organisms don't make their own food. They can't harness the energy of the sun. Okay, so it's the producers that are able to harness the energy from the sun and turn it into chemical energy in the form of sugar, which then our cells turn that sugar into another form of chemical energy, ATP. Okay. All right. So just remember what I said. That chemical reactions are going on in our cells all the time. Okay. But understand that most chemical reactions without catalysts, okay, will be too slow for there to be life, okay, for you to, if I want to say move my arm without catalysts, I want to be able to move my arm quickly enough the way I would want to, okay. And a lot of catalysts in our cells are enzymes, okay, so that's why, and enzymes are proteins, okay. So that's one of the reasons why enzymes are very important, is because without them catalyzing the chemical reactions in our cells, they will occur at too slow of a rate to be useful to us. Okay, so catalysts essentially speed up the rate of the chemical reaction. Okay. Without being chemically changed or becoming part of the product. So they essentially lower the activation energy. A lot of times, if you think about it this way, when you're pushing something uphill, it takes it's slower as you know, pushing something downhill. It's going to happen quickly. Think of a chemical reaction. I don't know if it's in the next slide. It's not. But think of the a chemical reaction as it first has to kind of go uphill and then it goes downhill. But catalysts, what they do is they make that hill smaller. Okay, instead of being as high of a peak, they make the peak smaller, so it's faster to get going. Because once it's going downhill, the chemical reaction is going to just go real quickly, essentially. All right. But again, that's why enzymes are very, very important, because they make the chemical reactions occur at a faster rate. Without enzymes, chemical reactions will occur too slow. All right, because catalysts increase the rate of the reaction without being chemically changed or becoming part of the product. And also understand, like we also need vitamins and minerals, okay? And a lot of those vitamins and minerals that we need for our body are coenzymes, okay? Meaning that they take part in these chemical reactions, and the enzymes need these vitamins and minerals for them to function properly. So that's why you have all these essential vitamins and minerals, because they still, a lot of them are coenzymes. So understand that's how they are important. All right, so just understand that because again your blood is mostly water. The cytoplasm fluid in this, which is fluid in the cell, is mostly consists of water. So water is a very good solvent, and things electrolytes can dissolve in water, okay? Like salt dissolves in water and it forms ions, okay? Those are electrolytes. Sodium becomes positively charged, chlorine negatively charged, okay? You see this sodium in the crystal, but once it's dissolved in water, it's gonna form ions, okay? Those are electrolytes, okay? And again, those are very important. You talk about, you know, we always say when you're dehydrated, you're sick. And you dehydrated, you drink electrolytes, Pedialyte for kids, and Gatorade is usually what people just say drink for when you're an adult or whatnot. But you want electrolytes to help rehydrate yourself, okay? And you also understand that, especially with your blood, and your cells too, you don't want. So when water dissolves, okay, you can also form H plus and O negative, okay, and then again, an acid when it dissolves, it's going to be at H plus. It's going to be in excess of H plus. And then when you dissolve a base, you're going to have an excess, meaning you're going to have mostly OH form, okay? And just remember with acids and bases in the blood, you don't want your blood doesn't want to be too basic. It doesn't want to be too acidic. Blood is naturally around 7.28, something like that. So it's very slightly alkaline. It's almost neutral, but a little bit slightly alkaline, okay? But you don't want it to become too acidic or too basic, okay? That's bad. It has to stay in a certain pH range, okay? There is also a YouTube link, okay, video dealing with acids and bases that you should watch also. All right, so this is just showing you a pH scale. So remember, 7 is neutral, okay? Anything lower than 7 is acidic. Anything greater than 7 is basic. So cytoplasm right around 7. Blood, slightly basics, 7.4. Urine is slightly acidic, 6.0. Coffee, some other things just to put it in perspective is pH around 5. Orange juice around 2.8. Then our stomach acid is very, very acidic, okay? And that our stomach acid being acidic aids in breaking down our food, okay? So that our small and large intestines can absorb the nutrients. All right, so the stomach has to break it down so that our small and large intestine can absorb the nutrient. So stomach is very, very acidic, stomach acid. So pancreatic juice, okay, around 8, so that's basic. And then ammonia, very basic, 11.9. All right, so that's just the writing, summarizing what I just said. All right, so you hear the word organic versus inorganic. Okay, so you hear the word a lot when you go to the grocery store. You hear the word organic a lot. Okay, what they're talking about at a grocery store is different from organic that they're talking about in chemistry. Okay, in chemistry, organic means anything that contains carbon. It's that simple. If it contains carbon, then it is organic. And essentially, all living things contain carbon. So all living things are organic. Okay. So at grocery stores, though, they use the word organic, really to mean all natural, meaning that they didn't add pesticides or they didn't add growth hormones, things of that nature. So in grocery stores, they're using organic really to mean all natural. Okay, it's almost like calling soap antimicrobial, right? All soap is antimicrobial. All soap, no matter what type of soap it is, can kill bacteria. Okay, so you're not lying. Like all food is organic. Okay, because everything living contains carbon, but essentially it's marketing. They're using it to say we didn't use growth hormones or pesticides on this food. Is really what they're saying. Okay, again, from a chemical point of view, anything that contains carbon is organic. So all living things are organic. Inorganic means it does not contain carbon. So things that do not contain carbon are going to be things that are not living. So inorganic compounds are non-living compounds like water, oxygen, carbon dioxide, certain types of salts. Okay. All right. So again, inorganic substance, water, the most abundant compound in living material. 2/3 of the weight of an adult human and it's a major component of all bodily fluids. Okay. Just know that with dehydration, what happens is like when you're forming a peptide bond, when you're forming a peptide bond, so you got these amino acids and you're going to link these amino acids together to form a protein. What you're doing is actually a dehydration synthesis reaction. You're removing water between the bonds and then you're going to form a larger molecule. And the way that a lot of the body does the hydrolysis reactions, when you're breaking things down, essentially you're adding water to these bonds and you're breaking the bonds by adding water to the bonds and you're creating smaller molecules. Okay, so synthesis and reactions tend to go by dehydration, removing of water, and then hydrolysis is the way the body tends to break down large molecules by adding water and breaking the bonds. Okay. All right, so organic substances. All right, so carbohydrates, very important. Monosaccharide, mono, that prefix means one. Basic unit of a single sugar. Okay, example is glucose. And again, monosaccharides, glucose is one of the main energy sources for the cells. And you can build on these monosaccharides to make bigger polymers. Okay, you can build on these monosaccharides to make polymers, and that's how you make starch. Starch is just a polymer of glucose. Okay, and then chitin, the shells of crabs and lobsters, those are also polymers. Okay. Disaccharide, the di means double. So the prefix di tells you that there's two. All right, then saccharide means sugar. So you have two monosaccharides are linked together to form a disaccharide. Example of a disaccharide is maltose. Okay. All right, so again, monosaccharide, one sugar, disaccharide, two sugars linked together. Polysaccharide, they're a polymer. Polymer means many sugar linked together, and polysaccharides are things like starch. Okay. All right, so polysaccharides, many single units linked together. Again, glycogen is also another example of a polysaccharide. Glycogen is a storage form of glucose, so storage form of carbs. All right. You also find polysaccharides in other natural materials. So cellulose, they're part of plants that you can't digest. All right, is a polysaccharide, a carbon polysaccharide. Chitin, again, the shell of crabs, lobsters, insects, another polysaccharide. And then starches are polysaccharides also. All right, so we talked about carbon. So now we're going to talk about another organic substance, lipids. All right, made of C, carbon, hydrogen, and oxygen. But lipids are fats and oils, and they have a basic unit called a fatty acid. Okay, so fats are lipids that are solid at room temperature, and oils are lipids that are liquid at room temperature. Okay. And fats and oils are hydrophobic, meaning that they do not dissolve or mix with water. Okay, hydrophobic. So here you go. Here's an example of a fatty acid. So you hear a lot of times the term saturated fatty acid versus unsaturated fatty acid, saturated fat versus unsaturated fat. And all that has to deal with is the double bond, okay? If it has no double bonds between the fatty acids that are linked together, then it is saturated. If double bonds are present, and this one has double bonds are present, then it's unsaturated, okay? All right, so remember dehydration, you're removing water, builds, okay? So a dehydration reaction builds a triglyceride. So for a triglyceride, you're gonna start off with glycerol. So you start off with glycerol, okay? And then you're gonna add 3 fatty acids to the glycerol. So you start off with glycerol, all right? Add 3 fatty acids to it, and then you have a triglyceride, okay? And you are removing water, okay? Triglycerides are used primary for energy, most common lipid in the body, okay? So again, the building blocks are glycerol, then you add 3 fatty acids to the glycerol, all right, and you have a triglyceride. All right, so phospholipids are you have the glycerol backbone again, okay? But this time you have 2 fatty acids and a phosphate attached to it, okay? So phospholipids are the main component of cell membranes, okay? So this is very important. So phospholipids have a water-soluble head. The phosphate group is usually the water-soluble head, meaning that it likes to interact with water. It's hydrophilic. But the tail is a fatty acid tail, okay, that is hydrophobic, that does not like water. So let me see if there's a slide. Here we go. Here go a good slide. So if you look at the lipid bilayer, this is why I call it a bilayer, because you have 2 layers of phospholipids. You have 1 layer of phospholipids here. You have 1 layer of phospholipids here. And if you notice, the hydrophilic heads interact with the water, but the hydrophobic cores, you see these tails, these hydrophobic tails, they interact with each other. So what happens is, so this is outside the cell, okay, and this is inside of the cell. So what happens is, you had the hydrophilic water-liking heads interact with the inside of the cell, and they also interact with the outside of the cell, but you have these hydrophobic tails that do not like water in the middle. So that's going to only allow essentially anything that's positively charged, anything that's large and has a positively charged, it can't pass through this hydrophobic middle of the lipid bilayer. Okay, so that's going to make the lipid bilayer semi-permeable. So only certain things, small non-polar things can pass through. Only small non-polar things like gases can pass through the lipid bilayer. Anything large in charge is not going to be able to get through this hydrophobic core middle right here, which is made of the fatty acid tails, okay? All right, so lipids, besides just being used for the plasma membrane or the cell membrane, you also have lipids or steroids. Steroids, the functional unit of a steroid starts off with cholesterol, and then you add things to it, okay? You add other things to it, and that's how you make different steroids, okay? Start off with a cholesterol, then you add things to it, and then that gives you, depending on what you add to it, the different types of steroids, okay? But just know that cholesterol plays, like we talk about how bad cholesterol is for us, because most of us get too much cholesterol from red meats and things of that nature. But your body needs a certain amount of cholesterol, because cholesterol stabilizes cell membranes. I don't have a picture of it up there, but let me just go back to the picture of this cell membrane. So you see how this cell membrane, you have the lipid bilayer. What cholesterol does is it's going to form it's gonna bond like right in here, okay, and it's gonna form little kinks in the lipid bilayer. That's gonna push it kind of together, like right in here, and it helps stabilize it. So remember, you don't want the lipid bilayer to be too fluid, but you also don't want it to be too stiff. So there's a good balance that you want for the lipid bilayer. You don't want it to be too fluid. You don't want it to be too stiff. So what the cholesterol does is it keeps it from being too fluid, from being too loose. It forms little kinks in between the lipid bilayers that makes it more firm, makes the lipid bilayer more firm. Okay, so that's how it stabilizes the cell membranes. All right, so other organic substance is proteins. Contains carbon, hydrogen, oxygen, and nitrogen. So the basic unit of proteins are the amino acids. All right. So again, proteins are very, very important for the body. Like I said, all enzymes are proteins. Okay. So, and also remember that proteins are formed on ribosomes. Okay, ribosomes in the cell are where amino acids are linked together to form protons through peptide bonds. Okay, but essentially, after an amino acids are linked together to form protein bonds, the protein has to fold. A lot of times it folds on itself, okay, and then it becomes functional after it's folded properly. And then also understand proteins are sensitive to heat, and acids or bases. So pH and heat, okay, if it's too acidic or too basic, can denature proteins. And then if they're denatured, they're not going to function properly. So temperature and pH play a very important role in making sure proteins don't get denatured. You don't want them too acidic, too basic, or too hot, or too cold, okay? So here you go, you got two amino acids joined together, a peptide bond, okay? So, but it's gonna be more than two, it's gonna be many peptides, many amino acids linked together to form a peptide bond, okay? Then you see how this peptide bond has folded on itself, okay? So it can be functional. All right, so again, peptide bonds are formed through dehydration synthesis. You should be used to this by now. Dehydration synthesis meaning you're going to link two amino acids together, and while in the process of linking two amino acids together, you are going to remove water. So water gets removed, okay, and that's how you form that peptide bond, okay? And when you want to break that peptide bond, you're going to add water to it to break that peptide bond. That's called hydrolysis, okay? So here's examples of primary structure of a protein is just the amino acids that are linked together by peptide bonds. All right, then the secondary structure are things like alpha, helixes, and then tertiary structure. So when they fold on each other is when they peptide folds on itself, and then the quaternary structure. So we have more than one polypeptide interacting with each other. Okay. All right. So nucleic acids are the building blocks of nucleotides, and you have two types of nucleic acids. The main two are deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. Okay. So just to tell you the differences between DNA and RNA, is essentially you have to look at the sugar. So for a nucleotide, you have a phosphate. Okay, both DNA and RNA are going to have a phosphate. You have a phosphate. Then you have a sugar. Okay, DNA has deoxyribose for its sugar. So this is the sugar right here. DNA has deoxyribose for its sugar, whereas RNA, the sugar is going to be ribose. Okay, so that they have different sugars. So DNA and RNA have different sugars. And then nitrogen bases. All right, so there are 4 nitrogen bases. DNA has adenine, thymine, guanine, and cytosine, and RNA has one different nitrogen base. So RNA has a adenine, but RNA does not have thymine. It has uracil instead of thymine. Okay, RNA still has guanine and cytosine. Okay, so whenever you see also another difference is but let me just say this: whenever you see, you're gonna see DNA and also messenger RNA written in triplets. So you're gonna be written in pretty much 3 nitrogen bases. If you ever see a U in that codon in that triplet, you know you're dealing with RNA. If there are no U's, if there's T's, you know you're dealing with DNA. Okay, when you're reading the triplets, the nitrogen bases, the codons. All right. Also, be aware that DNA is double-stranded, so it forms this double helix, which is kind of like a ladder twisted on itself. Think of a double helix as a twisted ladder. Okay, a ladder that's twisted on itself. And you see right here, DNA. You have your phosphate, sugar, phosphate, sugar, phosphate, sugar. So the sides. I'm sorry, I didn't mean to go off that slide. The sides of the ladder are the sugar, phosphate. Sugar, phosphate are the sides of the ladder, and the rungs, the middle part, the rungs of the ladder are the nitrogen bases, which are hydrogen bonding with each other. Okay, so you have the nitrogen bases that hydrogen bond with each other, and there's a certain matching of the base pairing. Okay, adenine always hydrogen bonds to thymine, and guanine always hydrogen bonds to cytosine. So if you look at this color coded, you see guanine, which is purple, is always going to hydrogen bond to the pink cytosine. Okay, and then the blue adenine is always going to hydrogen bond to the yellow thymine. Okay, so those hydrogen bonding between the bases always occurs. A hydrogen bonds with T, and G hydrogen bonds with C. Okay, and that's in the middle. The middle rungs of the ladder are where you have your hydrogen bonding between the nitrogen bases. Okay, and also another difference between DNA and RNA. RNA is single-stranded. So just in general, DNA is way more stable than RNA. RNA, one for 2 reasons. Okay, so DNA has a more stable sugar. Deoxyribose, this sugar deoxyribose is more stable than the sugar ribose. Okay, so that's one of the reasons. And then also DNA is double-stranded. Okay, whereas RNA is single-stranded. So essentially, you can, if you're working in a molecular biology lab, you can extract DNA and leave it on your workbench, all right, for a long time, okay, and without it degrading, okay. But if you extract RNA, you can't leave it out on your workbench, all right. It's going to start degrading. RNA is less stable than DNA. And also just understand that DNA contains the code for, it has all your hereditary information that makes your eyes blue or hazel or whatnot. But it also has all of the blueprints, the code for making proteins, okay? And understand your DNA never leaves the nucleus. Your DNA is very, very important, so it never leaves the nucleus. So what happens is you have messenger RNA. So the name messenger means that it must be carrying a message, right? So messenger RNA goes into the nucleus. It makes a copy of the DNA, but the copy is going to be in RNA, meaning that you're going to have uracil instead of thymine. So you see the uracil instead of thymine, you know you're dealing with RNA. All right. And then that messenger RNA can leave the nucleus and then go to a ribosome and that's in the cytoplasm, or the ribosome that's attached to the rough endoplasmic reticulum. Okay. All right, so that is the end of chapter 2, part 2 online lecture.